and welcome to the session of continuous virtual learning session today. The topic that I've chosen for today is biotechnology from the unit 4 of biology. The syllabus has actually been modified a bit. I'm sure you are all familiar with it. Just to give an idea that here the isolation of genetic material, that is DNA, this has been removed for this year. And earlier we had two vectors, two restriction enzymes, EcoR1 and Hindi3, Hindi2. But this year, these enzymes have been removed. Only EcoR1 remains. Now, similarly, from the methods of selection of recombinants, earlier we had three methods, but this year only two are left. Insertional inactivation method is deleted for this year. Mind it, only for this year. So, what is biotechnology? Actually, the term biotechnology was coined by Carl Erecke, and this was in the year 1919. And what a coincidence! This is the year 2020. So, how rhyming it is 1990, 2020. Just calculate whenever you are free how many years since then. So, he is regarded, therefore, as the father of biotechnology. And what he defined is the production of products from raw materials with the aid of living organisms. Here the word is living, that is more important, living organisms. The most acceptable definition, all comprehensive, all including definition, was given by the European Federation of Biotechnology, that is EFB. According to this federation, biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services. Now, here I'll remind you, if you see the list of abbreviations at the end of the syllabus, you'll find this word EFB as an abbreviation which might be asked as a question in the examination. Expand EFB, write the full form of EFB. So EFB, European Federation of Biotechnology, be sure that the spellings are correct. Next, a very simple definition, but a perfect definition. Biotechnology deals with techniques of using live microbes or their enzymes to produce substances, products, and to develop processes useful for the human beings. Now, when you see the examples like curd making, bread making, wine making, all these examples, it's not that these started after 1990 when Carl Erecke introduced the term biotechnology. Then only people started all these. This was long time ago, it was all happening. Therefore, let's draw a line between this traditional biology and the modern biotechnology. Traditional biotechnology refers to the ability of naturally occurring microbes to cause fermentation. So it was based on exploiting this ability. Traditional biotechnology exploits the ability of naturally occurring microbes to cause fermentation and to produce different products. Whereas in modern biotechnology, see, there is a very small change and very important change, that now it is based on genetic manipulation. Genetic manipulation, so that the results change according to our need. The products with a different flavor, different color, all these can be produced now. Earlier, we had to accept them as they were, but now we can make the changes according to our needs. So that is the role of the genetic engineering. In the modern biotechnology, now you can see there are two core areas, two main areas. One area is concerned with the genetic engineering, and the other one with the culture technology. It implies that first we make changes in the genetic constitution of the organism. This can be done by altering its DNA or RNA. Once we have succeeded in changing this, then we can introduce this DNA into a suitable host where the DNA will multiply, and then we can see the effects. If the change is desirable, there will be a desirable change in the phenotype of the host. So that is what we were targeting at. Okay, once we produced this, now remember, this was done in the laboratory situation. It was done in a flask, maybe in a petri dish, maybe in a test tube. Now that cannot be used at a very large scale. So to use it at a larger scale, what is done that once we get this genetically modified organism, so to say GMO, genetically modified organism, then this organism is now grown under strictly sterile conditions, germ free conditions where unwanted microbes do not grow. We have only the target organism growing. So, this is culture technology where we maintain a sterile ambience to enable the growth of only the desired microbe or the cell that was genetically modified in large quantities. This is the key, large quantities for the manufacture of useful products. These products can be hormones, vitamins, enzymes, antibiotics, anything. Now, for doing this, we have to follow certain processes, certain protocol. This protocol is fixed for whatever product we are going to produce. Whether you want enzyme or a hormone, or simply you are just wanting a copy of the DNA. So we have to follow certain specified processes. So these processes we have summarized here. So this is you can say the theme of biotechnology. All the biotechnological processes are based on this. By doing here and there small changes, you can get the desired product, but the basic theme remains the same. So the steps in the sequence, particular sequence, first thing, we isolate the target DNA. DNA which is coding for the product. So this DNA is also called as the foreign DNA, the passenger DNA, or simply the insert. So this DNA is actually part of the whole organism's genome, entire genome. When we are isolating, we don't have any technique to isolate only the small gene. Something like if you want to study a book, a particular chapter in that book, you can't take out only that chapter. You have to take the book, then from that book, you have to look for that particular chapter. So, similarly, this is the whole of the DNA, entire DNA. From this DNA, we need to find out the gene of interest. So, for this, we have to cut this DNA into smaller fragments. For this, we use a specific enzyme, very specific enzyme and very characteristic enzyme That is called the restriction endonuclease. There are a variety of restriction endonucleases available for doing this. So we isolate the DNA, cut it into fragments, then we isolate that gene of interest by a process called electrophoresis. Once we have obtained that fragment, the gene of interest, then we need to ligate this DNA fragment into a suitable vector which will carry it into the host. So we ligate the DNA with the vector and we get a recombinant DNA, rDNA. rDNA stands for recombinant DNA. Now this recombinant DNA is transferred into a host where it will multiply or it will be transcribed or translated as per the requirement. Then this host needs to be selected first from a population of non-transformed hosts. Once we have successfully selected the transformed host, then we culture it, we grow it in a nutrient medium for large scale production, from where the product can be obtained. Now we have to discuss each step in detail. But before that, this entire process that we discussed just now, from here to here, this entire process can be summarized in the form of this diagram. Here, suppose we take an example of human gene. It could be interferon, it could be insulin, it could be growth hormone, any gene. So we want insulin, we want interferon, or we want to produce the growth hormone by the process of genetic engineering. So that means that gene is our target DNA. The passenger DNA, the foreign DNA, or the insert, as I said. So this has to be ligated to a vector. So here this bacterial plasmid, you can see this is acting as a vector. Now comes the role of the restriction enzyme. Using the same restriction enzyme, this is the word, not just the restriction enzyme, the same restriction enzyme. You'll know why I'm emphasizing it. There is a reason behind, we'll know later. Same restriction enzyme is used to cut the target DNA and the vector. And now you can see here that the ends which are produced, both of these DNAs, these are complementary to each other. That is why we need the same restriction enzyme. That was the secret. So once these two DNA molecules from two different sources, when they are brought together, they hybridize. Hybridize by forming hydrogen bonds. But because hydrogen bonds are weak, so this DNA might detach from here. So just to make sure that it is permanently ligated, we have to use another enzyme, DNA ligase. So with the DNA ligase, now see, this enzyme has been now ligated to this. Now we have a DNA, you can see here this DNA. It's a colorful DNA, not exactly colorful, but this green color referring to the DNA of the vector, and this red color is indicating the target DNA. Now this DNA having two pieces from two different sources. That's why it is a recombinant DNA. It has been joined by combining DNA 1 and DNA 2. That's why recombinant DNA. That's why this technique is called RDT. R for recombinant, D for DNA, and T for technology, recombinant DNA technology. So this recombinant DNA now needs to be inserted, to be transferred into a suitable host. Just take care of one fact, that if it is interferon, this will be yeast. If this is insulin, this will be E. coli. If this is something other growth hormone, it will be human cell or chick cell, some other cell, animal cell, meaning thereby that the choice of host depends upon the choice of the target DNA, the type of target DNA that we are using. This will help us in selecting what kind of host we are going to use, which organism is going to be the host. So we transfer this recombinant DNA into the host cell, and now if the host cell is transformed, we identify the transformed host cell. This transformed host cell is now transferred into the culture medium, and in the culture medium it multiplies. Now there are multiple copies of the host cell. Therefore, this process is called cloning. And after that, once you get sufficient number of the host cells, take them out, break their cell walls from the cytoplasm, take out the product. In this case, suppose it was the yeast cell. From the yeast cell, the interferon was extracted, purified, and then injected into the patient. So you can summarize the entire process of RDT, recombinant DNA technology, in this kind of flow chart, diagram based chart, or you can go on writing the paragraphs and sentences. But you have to be very careful that if you are using this chart method, each and every step must be properly explained here. So we come to the first step of the process, that is the isolation of the genetic material, that is the gene of interest, the foreign DNA. And think about it, where you find the DNA in a cell. So DNA is a part of the nucleus, protected by the nuclear membrane, by the cytoplasm, by the cell membrane and outermost cell wall, if it is a eukaryotic cell. In a prokaryotic cell, what you'll find? You'll find that it is a cell wall and a cell membrane. There is no nuclear membrane, but still you have to cross these barriers to reach the DNA. So if we have to take out the DNA, we remove the cell wall. Depending on the type of the organism that we are using as a source organism, removal of the cell wall, the enzyme lysozyme can be used for the bacterial cells. Cellulase can be used for removing the plant cell wall, and chitinase for removing the fungal cell wall. For example, if it is yeast cell, you'll require chitinase. Thereafter, some detergent is required to remove the membranes. Now, whatever is floating is called as lysate. From this lysate, you have to take out the DNA. But this lysate is a mixture of biomolecules, a whole lot of biomolecules. The most important being proteins and RNA. And here, these are called the interfering substances. In the extraction of DNA, RNA and proteins are the interfering substances. So we have to remove these interfering substances by using specific enzymes, like use RNase, ribonuclease, to remove RNA, and use proteases to remove the proteins. Now you have only the DNA left in the medium, but still it is in a soluble state. So to precipitate it out, we have to use chilled ethanol. Ice cold ethanol is added into this lysate. This causes the ethanol to cause the precipitation of DNA. This DNA comes out in the form of fibers, and these fibers are spooled out as fine threads by using a fine capillary tube. Keep on rolling the capillary tube into the mixture. The DNA will wrap around it. It will all come out as a mass, white fibrous mass. So as we did earlier, here also the entire process we are going to represent diagrammatically to make it more precise. So you can see here, this is a test tube containing the cells. These cells are treated with the enzymes and the detergents to remove the cell wall and the cell membrane. Then the lysate, the cell contents, are treated with RNAs and proteases to remove RNA and proteins respectively. Then we add ethanol, the chilled ethanol. Chilled ethanol causes it to precipitate, and this is now spooled out on a capillary tube. Now this DNA is suspended in a suitable buffer, and then we process further. Further steps are carried out. Now we have the whole DNA. We have to get the fragment of interest, the gene of interest. So for doing this entire natural DNA, we call the native DNA. This native DNA has to be cut into fragments. And these fragments are produced due to the action of the enzyme restriction endonuclease. These fragments have to be separated so that we can find out the fragment of interest. And this is done on a principle called electrophoresis. This works on the basis of presence of electrostatic charge on the biomolecules like proteins, DNA, RNA. If they carry charges on them, these can be placed in an electric field, and by applying a charge, these can be separated. They'll separate according to their charge under the influence of that electric field. So this process of electrophoresis, it needs a matrix, a ground substance, that is agarose. That's why it is more appropriately called agarose gel electrophoresis. As we know that DNA is negatively charged, you can ask yourself, what makes the DNA negative charge? And try to answer yourself. So put the question and give the answer. So what produces the negative charge? Okay, I am giving you the answer. The answer is presence of phosphate groups in the DNA. This phosphate makes the DNA negatively charged. Therefore, the DNA can be separated by placing it in a mild electric field, whereby they will move according to their charge, not only charge, also their size, because now we are using the agarose, which will act as a molecular sieve, the sieving material. Gel will act as a sieving material. So this agarose gel, this can be used as a matrix which causes the DNA molecules to separate on the basis of charge, and to supplement it, to complement it, we are using the charge also. So size and charge, a mixture of size and charge. DNA is separated according to this process, and the theme here, the principle here is that smaller the fragment size, the faster it moves. So therefore farther it goes. Because the sieve, the gel, doesn't allow the DNA to move freely. There is one problem, that this DNA cannot be seen with the naked eyes. So to visualize this DNA, we have to stain it. Stain it with ethidium bromide, ETBr. It is abbreviated as ETBr. This ethidium bromide is a fluorescent dye. It doesn't have its own color. But on exposure to UV rays, ultraviolet rays, it starts emitting orange light. So these fragments can be seen in the form of orange bands. Once we have got the band of interest, we know this is the target DNA, then this is cut out from the agarose gel and extracted. This process of extraction of the DNA from the gel, this is called as elution. Now, the DNA that we obtained, this can be used for constructing recombinant DNA or any other process that we are going to follow. This is the electrophoretic setup. In a glass trough, buffer, and this is the gel, the agarose gel. In this agarose gel, there are wells. We call them electrophoretic wells. Holes are created, and in these holes, we load the samples, transfer the samples. Now the samples will start moving once we apply the charge. And take care, DNA being negatively charged is moving towards positive pole. So DNA moving towards anode. Once it has moved, we don't see the DNA moving. Even if we have added ethidium bromide, we cannot see it with the naked eyes like this. So we will have to expose this gel plate to UV rays. And on the exposure to the UV rays, now you start finding the bands here, orange color bands. From this, can you conclude something? Which one is the heaviest? Which one is the largest? Which one is the heaviest? Which one is the largest? So see at this. This one has moved farthest. So this one is the smallest fragment. And this has almost not moved at all. This one is the largest fragment. So largest fragment shows least movement, and the smallest fragment shows maximum movement, fastest. Now that we have got the DNA, now we have to join it with a foreign DNA and to create a recombinant DNA. So what are the steps of construction of recombinant DNA? The recombinant DNA, it was first of all produced by Cohen and Boyer. These are the people who started the genetic engineering in a proper way. So they created, Stanley Cohen and Herbert Boyer created the first recombinant DNA in the year 1972, sorry. So this, since then, lots of improvements have been made in this process. What are the tools of recombinant DNA technology? So there are two major tools, and these two major tools are, one, the restriction enzyme, which is acting as a molecular scissors, and the DNA ligase, which is acting as a molecular glue. So restriction enzyme, it cuts. So that's why the molecular scissors. DNA ligase, it joins. That's why the molecular glue. So it is as simple as cut and paste. Cut, restriction enzyme, paste, DNA ligase. For this, let's know something about the restriction enzymes. Restriction enzymes, also called restriction endonucleases. These restriction enzymes, these are naturally obtained from the bacteria. So bacteria, the natural source. From different bacteria, these restriction enzymes are isolated. So in the bacteria, they are meant to protect the bacteria, protect them from the action of bacteriophages. So as we know that viruses, when they infect bacteria, they inject their DNA. Once the DNA goes into the bacterial cell, this DNA is broken down into fragments by the restriction enzyme of the bacteria. But there is a condition that this bacteriophage DNA, this one, the DNA of the bacteriophage must possess a particular site. Then only it can be cut by the restriction enzyme, otherwise not. So each restriction endonuclease recognizes and cuts only at specific nucleotide sequences. So that is why not all the bacteria are spared. Some bacteria protect and some bacteria cannot protect themselves. So this is just because the restriction enzymes have specific sites, and these sites are called restriction sites. These can be recognized by their palindromic nature. We will talk about the palindrome in the DNA in the next a few slides, but this is a clue that the sequences are identified by their palindromic nature. Once the restriction enzyme finds the palindrome, then it cuts the DNA. See how it cuts. It cuts it like this. The restriction site recognized by restriction enzyme. And now you can understand what is a palindrome over here. That these are strands of DNA, 5 prime, 3 prime, 3 prime, 5 prime, the complementary strands. In this DNA, the sites are GAA, 5 prime. So the palindrome is not on this DNA. It cannot be that GAA, AAG. Think a palindrome like this. Palindrome has to be on the other sequence in the reverse order. As it was on this strand, in the reverse order on the other strand. Then only these two sequences, they become palindromes of each other. So GAA, 5 prime on this, and the other 5 prime, it is GAA, G and A. See here, the cut is produced here, G and A. 5 prime GA, this is the target site. So once the restriction enzyme moves along the DNA, it scans the DNA, finds the site, then it cuts G5 prime GA. Now it cannot cut it across like transverse section because this is not 5 prime GA. On this strand it is 5 prime GA. So on the other strand, on the complementary strand, this will be 5 prime GA. So the cuts are made here in the one of this strand and here on this strand. So the two sides, after these are cut, the phosphodiester bonds are broken, then this G and this A will separate, and this one here they will separate, and this is what the DNA will look like. G and A you can see separated, this G is separated, and AATC sequence, this has gone away. Here CTTAA, and this G, this has gone away. So there is a gap produced here in the DNA, this gap. Another interesting observation is that this GC paired, but this sequence TTAA, this remains unpaired. We call this overhang. Like in this strand, it is TTAA. This is an overhang of 4 strands. So this strand, this extended strand, this makes the DNA sticky. We call these two as the sticky ends. Sticky ends, they have the unique property that if they get the complementary sequence from any source in any DNA, then they will immediately hybridize, form complementary bands, hydrogen bonds, and these hydrogen bonds will again cause formation of a double-stranded DNA. So that is how it acts. this sticky end, this is a great help. Once you get the sticky ends, now see the entire process follows like this. The sticky end in one DNA, because you have used the same enzyme, so you are getting the sticky ends in the other DNA, and then the two strands are going to be complementary to each other. So when this DNA and this DNA from two different sources, when they are brought together, see what has happened? These two have hybridized, hydrogen bonds are formed, but still the bonds are weak. The gap is filled, of course, but it is not stabilized properly. This stabilization is done by another enzyme, ligase, the DNA ligase. So DNA ligase forms the phosphodiester bond between this G and A here and G and A here. So we get a complete double-stranded DNA. This we call the recombinant DNA. This recombinant DNA is now ready to be used for any chemical biogenetic experiment. Now there are a variety of restriction endonucleases, as I told you. Several hundreds of them are available. So we have to know them. We have to give them names. And there is a specific system of nomenclature. That system of nomenclature is uniformly followed all over. So whenever a restriction enzyme is discovered by a scientist, the scientist has to give it a specific name. As we know that whenever a scientist discovered an organism, a new organism, then they have to give the name, and that was a binomial nomenclature in the biology. In the previous class, you must have read about binomial nomenclature, like name of genus, name of species, italics, all these. So similarly, here also there are specific rules for nomenclature of restriction enzymes. So let's take an example of the EcoR1. This is a restriction enzyme, EcoR1. Here, E, the first alphabet E. It comes from the name of the genus of the bacteria. This E. coli is found naturally in the bacteria Escherichia coli, from where it was obtained. So this E refers to the genus of the bacteria Escherichia. So first letter comes from the genus. Then next two letters, as you can see here, ECO, CO. So this CO stands for the first two alphabets of the coli, Escherichia coli. So ECO, Escherichia, E, co for coli, Escherichia coli. Fourth alphabet refers to the strain, because the bacteria, they are found in different strains, varieties. So the strain RY13 was the one from where this enzyme was isolated. So RY13, the alphabet R is used, EcoR. And 1 is a numeral, 1, 2, 3, 4, so on. So this numeral, this number, Roman numeral, this refers to the order of discovery, isolation of this enzyme from this particular strain. So from Escherichia coli RY13 strain, this was the first. If there is a second, then from the RY13, then it will be called a EcoR2. Then from the same strain, if we isolate another one, it will be called as EcoR3, 4, 5, and so on. Similarly, let's take an example. Another one, HindD3. This is also a restriction enzyme. Now, first thing that I wanted to remind, that this is not HindD3, not HindD3. When we are reading it, it has to be read like HindD3, HindD3, not HindD3. So H for Haemophilus, the genus of the bacteria. In for the influenzae. So Haemophilus influenzae, the strain D, RD strain, third in the series. So from Haemophilus influenzae, the D strain, this was the third restriction enzyme to be isolated. So that is how we give the names to these restriction enzymes. The next topic will be the cloning vectors. And before I actually start talking about the cloning vectors, just a reminder that this is not a vector as we think of a mosquito, housefly, or a cockroach, the carriers of the disease. Not that. In recombinant DNA technology, we have to say very clearly that these are the cloning vectors, not simply the vector. No ambiguity. There shouldn't be any ambiguity. So it is a cloning vector. So cloning vector is not an organism. This is actually a piece of DNA, a double-stranded DNA, which is used to transfer the foreign DNA into the suitable host. So cloning vectors, we have a variety of cloning vectors. Depending on the size of the DNA, depending on the product, all these things decide which vector we are going to select. So DNA from which source? Whichever source we are going to get it from, it must have certain features. So any cloning vector has some features, what we call minimum essentials. So any cloning vector will have these three features, which we are going to discuss. The features are features of an ideal cloning vector. These are that it must have an origin of replication. The first and the foremost important point, origin of replication. This is a sequence from where the replication of the DNA will start. So if we link the DNA, foreign DNA, to this site, to this origin of replication site, then it can easily multiply. Otherwise, if we pick a DNA randomly and insert into a host without an origin of replication, it will be just lost from the system. Moreover, this site, this origin of replication, this also regulates the copy number of the linked DNA. Next point, next requirement in an ideal cloning vector, it must have a selectable marker. This selectable marker helps in identifying that which host has actually picked up the foreign DNA. So it is literally a marker which helps in marking the transformed host, the selected host, so that it can be used for further processes. And it must have cloning sites. More the better. These cloning sites are also called restriction sites. We have been talking about this. So multiple cloning sites, ideally, for different restriction enzymes, different cloning sites. So cloning site or the restriction sites are the sites which are recognized by the restriction enzymes and cut to produce the sticky ends, preferably sticky ends. In the nick thus produced, the foreign DNA can easily be ligated. We'll take one example of such vector. The vector is PBR322. There are several vectors which are mentioned in the course. If you refer to the syllabus, we have the plasmid-based vectors, the virus-based vectors, and artificial chromosomes. Each vector has its own advantage. So first one, the PBR322. So it is P for plasmid. This is a hint. P plasmid. This plasmid has actually been obtained from Escherichia coli, and then necessary modifications are made to make it suitable for our biotechnological experiments. So this plasmid was developed by Bolivar and Rodriguez. That is why BR. So plasmid developed by Bolivar and Rodriguez. So PBR. The features, as we discussed, the ideal features. So let's look for those features where they are present in PBR 322 or not. First feature, the size is very small, relatively 4,361 base pairs, 4.3 kbp, kilo base pairs. The advantage of this small size is that it does not break easily and can be easily taken up by the host bacterium. Second point, it has a bacterial origin of replication, which makes it easy for the foreign DNA to replicate along with this plasmid. And it has many sites, many unique recognition sites for different restriction enzymes. And as a selectable marker, there are two genes. One we write as AMPR and the other one we written as TETR. It stands for ampicillin resistance and tetracycline resistance. So these provide resistance against antibiotics so that the host can be selected. Like this. You can draw a diagram of PBR 322 and try to label its different parts. This could be a self-test. The sites have to be very, very specific. You cannot mark them randomly. Like where 7 o'clock strikes in a clock. That is the origin of replication. This point, origin of replication. And at 6 o'clock, there is another site ROP. This origin of replication, this is the site from where the replication starts. So it has this feature, origin of replication feature. Selectable markers, you can see there are two selectable markers. One shown in orange color here and the other in the blue color here. So this marks the position of the gene coding for some enzyme, beta-lactamase, that will metabolize ampicillin. So this gene confers resistance against ampicillin. This one provides resistance against tetracycline. These alphabets, PST1, PVU1, and so on, these are the ones which are the sites, restriction sites. So that means if we can use EcoR1, it will be cut from here. If you use BAM-H1, it will be cut from here, SAL1 here, and so on. Another example of a vector, the bacterial artificial chromosomes. Artificial chromosome because these are all based on bacterial chromosome but manipulated according to our demands. So BAC is based on F plasmid of the bacterium E. coli, Escherichia coli. Developed by Mel Simmons. Features you can see it has the replication site, multiple cloning sites, selectable markers, and this is a unique feature, PAR A and PAR B. This is not a compulsory that you find in all of them. So this is unique to BAC. But these essential features you can see always required in a vector. Here also these are present. Similarly, can be represented with the help of a diagram. The origin of replication, the selectable markers, so many, you can see a whole lot, so many restriction sites. So make it very useful. And that is why it was used. You must remember it. It was used for Human Genome Project. Yeast artificial chromosome, as the name tells you, yeast for eukaryote. All these till now that we discussed, these were for the prokaryotes. This one is for the eukaryote. So here there are minor differences. The initiation site named as ARS, and because eukaryotic chromosomes have telomeres, so we have to provide telomeres and centromere so that during cell division it is not lost. URA3 and tryptophan 1. These are the selectable markers, and multiple restriction sites as we have for the many vectors. See here, the telomeric ends, the selectable markers, and the restriction sites. This is how it is used. Cut it from here using a particular restriction enzyme, and in the cut, in the nick that's produced, insert the foreign DNA, and now you have a linear DNA, linear. Eukaryotic chromosomes are linear here. Next one, the agrobacterium. Agrobacterium, this is different in one sense that this is used exclusively for the plants. And it causes a disease called crown gall disease in the plants. Here it has a unique plasmid that we call Ti plasmid. Ti stands for tumor inducing plasmid. And this Ti plasmid, this has tDNA. After this tumor inducing region is removed, it is called tDNA. So tumor inducing DNA. This is the diagram of it, you can see. Origin of replication. The tDNA, this is where we insert. There are lots of enzymes and hormone producing regions. This is the virulence region that causes the crown gall disease. Then the last vector is retroviral vector. Retroviruses, RNA viruses, they can be used because they have the reverse transcriptase. So if we use reverse transcriptase, the viral RNA can code for viral DNA. This viral DNA can be integrated into the host genome, and then if we infect any host cell with this virus, that viral DNA will be transferred along with the foreign DNA into the host, and it will multiply. They are especially useful, note it, in the studies concerned with the oncogenes, and they have also been used in gene therapy. So with this, we come to the end of it. But before you start question answer or something else, this is a takeaway from my side. Do these assignments at your convenience whenever you get time. So these assignments, these can be done anytime at your convenience, whenever you feel like doing it. Thank you. Now I welcome the questions and answers from your side if you have any. Okay, so here comes the first question. What is the advantage of genetic engineering over the traditional hybridization procedures used in plant and animal breeding? Okay, in traditional procedures, we are just doing it randomly. We are just mixing them. We are relying on meiotic recombinations, the genetic recombinations. We are not sure which gene is getting transferred and which gene is getting left out. Whereas, if we are using genetic engineering, we know what actually we are doing. We are cutting the target and then transferring it. Okay, so next question has come. Why is it necessary to provide an ORI site? So ORI site, the site of origin of replication, it is called site of origin of replication. If we, as I discussed earlier also while doing the session, that if we take a DNA randomly from outside and throw it just into the cell, this DNA will not replicate because it doesn't have that ORI site. We know that DNA replication starts only at a particular sequence. So it will be left out. That's why it is necessary to provide the ORI site. Any other question? Okay, what will happen if a vector has more than one recognition sites, okay, for the same restriction enzyme? Oh, it will create havoc. It will cut the DNA into so many fragments. At each restriction site, it will cut. So we'll get not just one fragment, one opening. It will be just cut into smaller pieces, diced, so to say, as we do. So we don't have to do that. We don't have to cut the enzyme, the DNA at many places. We know where exactly we are cutting it at, so that we can insert the DNA precisely at that place only. Any other question? Oh, what is the difference between endonuclease and exonuclease? Good one. Endonuclease, as the name tells, nuclease, let me tell you first, nuclease means the enzyme which cuts the nucleic acid, and here the nucleic acid is DNA. So this nuclease will cut the DNA not at the extreme ends, somewhere inside. The phosphodiester bonds will be cut inside the molecule, producing smaller fragments. Whereas exonucleases cut the DNA at the terminals. You know that DNA has a 5' end and a 3' end. So if it is cut at 5' end, we call it a 5' exonuclease. If it cuts at 3' end, we call it 3' exonuclease. So exonucleases release single nucleotide one by one, not fragments. Oh yes. Does any restriction in nucleus produces blunt end? Good one. Yes, there is a restriction in nuclease that is called HIND2. Another one, EcoR5. These are the ones which produce blunt ends. So it's not very easy to use them, but with certain modifications, these have also been used in genetic engineering. For this, we use certain extra assistance of terminal transferase enzymes. That's altogether a different story. Any more? Southern blotting. Oh. We actually I use the term elution, eluting the DNA from the gel. That was called elution. Now, this technique of eluting the DNA from the gel was developed by a scientist by the name of Southern. That's why it is called as Southern blotting. So, the process of extraction of DNA from the gel, just one liner, process of removal of DNA from the gel is called as Southern blotting. Reporter gene. Reporter gene, literally, these are the reporters. They reach at the site and report that, okay, I have reached. That is the analogy. This is actually, reporter gene is actually another name for the selectable marker. Selectable markers because they help in the selection of the transformed host. So they are also called the reporter genes because they report that, okay, this is the host that has actually been transformed. Any other? What is the advantage of using agarose? Okay, agarose as the matrix in the gel electrophoresis, because one thing, agarose, we can control the pore size by using the degree of hydration, so that if you want only the smaller DNA to move, we can control that thing. But even more important is, the foremost important reason is that agarose itself is non-reactive. It is an inert material. It does not react with DNA or ethidium bromide or any other chemical that we are using in the medium. So due to its inert nature, agarose is preferred. Oh, retroviruses are usually carcinogenic. That is correct. How can they be used? I would have loved it to answer it some other day also, but no problem. Other day it will come naturally, but today also we can discuss it, that carcinogenic, but still it is used. So before using this carcinogenic or tumorigenic, we have to remove the cancer causing gene. So, in the terms of biotechnology, if I use the term, this is called disarming. So, we don't use the retroviruses as such as they come. No, we have to disarm the retrovirus. So, how can they be used? They can be used by disarming. After disarming, it can be used as a normal vector, as a peaceful, as a good vector. Thank you. I believe this is the last question. So, thanks a lot.